Thanks everyone for being here. My name is Kristen Hood. I'm the Executive Director for Research Engagement at NephCure Kidney International. We appreciate you being here today. Josh Tarnoff is going to do some introductions of the Pfizer group. However, before we get started, I just wanted to let you know that at the end of this presentation, we are going to open up the forums to allow you to ask questions to the Pfizer representatives that are here today. The best way to do this in a webinar situation is if you wouldn't mind raising your hand, and I can allow you to talk when the time comes. The other option is for you to put your question in the Q&A section of that Zoom toolbar. Either way, I'll be able to get to you. I will be moderating the Q&A at the end, but we would love to actually hear your voice and you actually ask the questions personally to the Pfizer group. So when it's time, make sure that you have that button ready to raise your hand. And Josh, with that, I'm going to turn it over to you.
Thank you, Kristen, and welcome. Good afternoon, everyone, and thank you for joining this very, very special program this afternoon. So again, my name is Josh Tarnoff. I'm with NephCure Kidney International, and I want to thank our hosts from Pfizer. Before we begin, real quick about NephCure, for those of you who aren't familiar. So we are a patient advocacy organization dedicated to supporting the patient all through their journey with nephrotic and nephritic and practically all rare protein spilling diseases. And we're a little bit different from a lot of organizations in that we have a very strong research orientation, and our commitment to not only supporting the patient, but the best thing we can do for any patient, of course, is to find a cure, right? And so our mission over the past 20 years has been engaging with basic research, translational research, and of course, most recently with the onset of all the beautiful clinical trials that we've seen. I think we're up to almost 40 nowadays. I want to thank Pfizer, who's been with us for a long part of that journey. And in our history, we've dedicated and committed over $40 million worth of research through the years. And you may have seen us engaged, of course, with Neptune and the CureGN initiatives, as well as a whole bunch of other things. And as we got along the way for clinical trials, this initiative called Gateway years ago, where the goal was to remove the barriers to clinical trial recruitment and to ensure that every trial successfully recruited. So our pedigree is long and deep with research, and so we're honored today to partner with Pfizer in this incredible initiative that really looks at continuing that tradition of looking for what's new, how do we support what's coming out, and we want to thank Pfizer, who's been a big partner of NephCure's for many years in that endeavor. So with that, I'd like to introduce our host today, Steve Barazzi from Pfizer. Steve is the Senior Director at Pfizer's CTI. We'll learn what CTI is in a minute, right? Who will talk about Pfizer's collaboration on Robo, in particular, its association with Boston University. But before Steve joins us, Aileen Healy, who's the Executive Director and Global Head of ES&I for Rare Diseases at Pfizer. Boy, that's a long one. I guess you've been there a while. They keep adding titles to you, right? So through the CTI, which is the Center for Therapeutic Innovation. So thank you both. And Aileen, you'll be talking, I guess, about the many ways that folks can partner with Pfizer, including CTI, and sharing a little bit more about that. So with that, I thank you both once again. Thank you, Kristen, and I hand it over to Aileen. Thank you.
Thank you very much, Josh and Kristen. Appreciate that and appreciate everyone's attention today. We're excited to have the opportunity to describe for you how Pfizer looks to advance the most innovative science through partnering. And we're going to describe for you what a lot of our acronyms mean, and we'll try not to use too many. It seems to be a habit at Pfizer. But we really want to describe for you how there is a lot of ways, and Pfizer is flexible in how they look to partner with people. And in order to do that, I think we want to, spend a little bit of time explaining the organization. It's a big place, and I know that we've been in the news a lot lately. And if I can figure out how to advance my slide here. I think the point of the discussion today is really to understand how, a company as large as Pfizer, who, for example, has treated over 1 billion people with the vaccine, or has treated, tens of millions of patients for cancer and cardiovascular disease, how we think about developing treatments for rare renal diseases and for renal diseases in general. I'm focused in the rare diseases specifically, but I have colleagues who are also in our internal medicine group, and chronic kidney disease is a focus area. And I'm going to walk you through sort of how we're organized, what our focus is, what some of the strategies are. Hopefully it won't be too dry in going through sort of how we're organized to understand what we're looking for. And we hope that this is an interactive discussion. And then, we'll describe for you at the end of my part a call for proposals that we have starting next month that we're really excited about for rare renal diseases. And Steve has been instrumental in that. And we thought it would be really helpful then to give you an example of a program that started from a call for proposals and is now in clinical trials. And it's really exciting to see how that's advanced. And I know Steve has really just done an amazing job in that, and it's a good example of what a CTI program can do. So let me just run through this, and please, any questions now or at the end would be welcomed. So Pfizer is organized as an end-to-end company, and what that means is internally we start at ideation and go all the way through clinical development, through approval, registration, launch, and marketing. And you can see, although we've been in the news for the vaccine and the team who've worked on that have done an incredible job in advancing that so quickly, we have almost 90 programs that are in clinical development right now. And those come to Pfizer either through our internal efforts or through partnerships throughout the whole pipeline. And so I want to explain a little bit more about the way we think about that. So we're divided into 5 therapeutic focus areas. Oncology is our largest research unit, internal medicine, where we have a focus in chronic kidney disease, inflammation and immunology, the rare disease where I sit, that's the smallest research unit, and again, our vaccines unit, that's been in the news a lot. And I'm going to come back to this concept of breakthrough medicines and first-in-class science, because those are very high bars in drug development. And it's really important to think that, or to understand that we're really focused on these concepts. And there are a lot of, there are multiple paths to get to successful drugs. We have a real focused area that we're committed to. And so, we maintain an innovative world-class pipeline. And again, the way we do that is through internal efforts, but also sourcing the best available science externally. And that's why we're here talking to you to share with you what we're looking at. So hopefully we can advance some ideas maybe that people have or are working on in their own laboratories. So in addition to those research units that we have partner lines that support them, like medicinal chemistry, drug design, drug safety, but we're a separate department. Stephen and I and Jenna are in emerging science and innovation. And again, this is solely focused on external partnering and translating those partnerships into breakthrough therapies. And so it all starts with discovery, again, identifying and sourcing differentiated targets and biology. That's really, our day-to-day, what we're focused on, novel targets and biology. And the way we can advance that into first or only class medicines, is partnering, where Pfizer can bring their capabilities, deep expertise, and their resources to really rapidly translate that towards developing medicines. We also have ways to identify cutting edge science that may not be target ready yet, and de-risk that through collaborations, through seed investments, enabling work to advance, always looking towards the goal of breakthrough medicines. So these are sort of our buzzwords around first in class and differentiated resources that we're focused on. So just to drill down a little bit more within those 5 therapeutic areas that I described for you, they're broken down here into specifics. So I mentioned I'm embedded in rare diseases. We have over 40 rare diseases, mostly monogenic diseases, that are within strategy for us. And they're defined within these sort of 5 buckets, these topics listed here. And renal disease is relatively new. I've been at Pfizer for almost 5 years now, and it's really the last few years that this has come on the radar radar for our rare disease unit, and it's largely through this Centers for Therapeutic Innovation, or CTI, program that started, I think, almost a decade ago, that Steve's going to tell you about. And so you can see for yourself sort of, each of the research units has their focus areas. And then within our department, we're always being tasked with identifying what emerging science is out there that we can either support or bring in and leverage internally ourselves that will be sort of the next wave of advances in therapeutics for patients. And so we have 3 listed at the bottom of this slide I'm showing here, but we're constantly in our department, scanning the meetings and publications to really understand when things are starting to gel, where they may be actionable from a Pfizer point of view, for example. And we're very much focused on not only in our end-to-end setup, but in leveraging capabilities that we build. And I think the COVID vaccine and the mRNA technology that made that available is a really great example of how we do that. So we have a vaccine group prior to that, very focused on this mRNA technology, as it was, a novel approach to developing vaccines before COVID struck. And then, kudos to the team at Pfizer who, did a remarkable job working at light speed, I think they describe it as, to get that out to the public, really. But so now we leverage that internally within our vaccines group for flu and shingles and for other infectious diseases. And now it's starting to go more, beyond just vaccines into our therapeutic areas. Rare diseases is, fit for purpose with monogenic diseases. One example, recently we announced a collaboration with Beam Therapeutics for base editing. We're also looking to apply that to vaccines for cancer. And then, that will continue to expand into other areas. So that's just sort of an example how we build a capability, and then we work to leverage it and maintain a leadership role. And, we do a lot of that through partnerships. That was a partnership with BeyondTech back before I think any of us knew what COVID was. And so, the different we describe them as, partnering or funding vehicles. So I think long before I joined, Pfizer realized that they needed to be flexible if they wanted to find the best science. And so we have listed here some of the ways that we do that. In the upper left, you see joint drug discovery. That really describes how we do our partnerships with our Centers for Therapeutic Innovation, or CTI. Spend a lot of time, identifying academics doing really interesting work that's matches or supports or advances our own internal work. And we put together research collaborations. These are collaboration agreements. They're really partnerships where we fund very defined work in someone's lab, for example. These are not grants. Pfizer doesn't really do the grant model. I think we found that partnerships are the fastest way to advance innovative science, where we can offer in-kind support with our own strength and partner with people who have, scientific expertise or technical expertise in an area. And then we also have sort of standard, business development, where we license or option or do merger and acquisitions to partner with people or bring in the best science. And then, as I mentioned, venture and seed investments, and I can describe that a little bit more. So, sort of one more operational slide about how our department, Emerging Science and Innovation, or ES&I, is set up, and you can see really these 5 functions drive our collaborations, our external collaborations, and help deliver value for Pfizer. That's really our goal. And, we're always tasked with finding emerging science, novel science, and, not waiting for it to come to us. So, this is a great opportunity for us to sort of, let more people know what we're interested in and look for potential new collaborations with folks. And so, my role is, the banner is across the top there, the emerging science lead. And so, not only am I embedded in the rare disease unit, and I have a counterpart in all of the research units and also for all the technologies, but we work very closely with our target sciences, which is, I'm going to tell a little bit more about that, our partnering group, and then our Centers for Therapeutic Innovation. And then, lastly, I mentioned we leverage our successes. With the success of COVID, we now have, since last summer, an RNA incubator, and that's really focused on, accelerating Pfizer's leadership in RNA therapeutics. So, just going to continue to drill down on, what we're seeking is early novel science, cutting edge technologies. And that's really, the focus of our role here. And so I just want to tell you a little bit more about these individual departments within that were, in addition to the Centers for CTI call for proposals, or, there can be partnerships with these other efforts within emerging science and innovation. And the first is target sciences. And, I would describe target sciences as sort of the de-risking that you would do before a program would enter the portfolio at Pfizer. So a lot of this is early stage biology, it's target identification, it's generating target hypotheses, and then it's doing the validation work and any, what we call confidence and rationale, really proof of concept before it would become a portfolio project. And the focus there is really on cutting edge human genetics, computational biology, and functional genomics. It's really linking mechanism with the clinical path of patients and trying to bridge that gap and understand how you could intervene at a target site to really change the course of a patient's disease. And so in addition to this sort of functional genomics and genetics capability, very strong in-house biology using genetic models of disease, both in vitro in patient cells and in genetically engineered models. And we'll work with academics, for example, who've developed a unique model that's not generally available through sort of CRISPR knockouts and things like that. And then lastly, confirming the target, and we often spend a lot of time doing that. Confirming findings in an independent lab is key to all science, and we have a group that really works through that at Pfizer. And so I mentioned sort of, the state of the art, what we're looking for here, human genetics. In the rare disease unit, this is especially important for monogenic diseases where clinical paths are not clear. Patients' databases, biobanks, for example, where we can, do omics studies to try and understand that link between disease mechanism and the clinical path, and also for discovery and validation. So those are the types of topics that we look for externally to partner with people and bolster the science really with some of our own internal capabilities. And then in addition to sort of the discovery and validation, we also have a diagnostics group. I'm not going to spend too much time on that. We don't seek external diagnostics, but we do have companion diagnostics with all of our clinical trials to stratify patients or for outcome measures to look for drug efficacy. So that's also an internal focus. And then partnering innovation, I'm going to go through this quite briefly in the interest of time, but I do want to tell you about Pfizer Ventures, for example, a separate venture arm, again, strategically aligned with the 5 research units that I described for you. And really, again, looking for innovative science. They invest across all stages from very early seed stage through clinical, but 80% are focused on therapeutics. So it's really about delivering breakthrough science for patients. The way Pfizer Venture works is they generally acquire a minority stake. They don't lead investments. They're usually part of a syndicate, and they'll be involved in leading the company through their own. It's a very experienced group, so either a board seat or an observer seat. And then the returns for Pfizer are either a financial return or more frequently now, companies being partnered or acquired that we've worked with in this sort of venture arm. We also have a medicines initiative. This is based out of the UK, largely works in the EU and the UK. And this is really public-private partnerships to advance, really unmet needs in health. And these are a very broad range of topics, but we have a long history in that, over 65 pre-competitive partnerships that we've done. These can be anywhere from, natural history study, clinical trials to, assay development. It really runs the gamut there. And we have, a focus team in the UK that's driving that effort. And then why we're here today is really to talk about Centers for Therapeutic Innovation. I'm just watching my time here. And we're going to describe a little bit for you, how this is organized. It's over 10 years old now, 10 or 12 years. And really the mission at the outset was find the best science. And that's evolved in the last few years to really find the best science that's strategically aligned with those research areas that I described for you that fall under our research units. And this is a unique collaborative model in that the academic investigator is paired with a scientific champion. In this case, Steve Barassi is going to tell you how he did that. And what the partnership there is taking advantage of is the scientific expertise and the findings of the academic investigator and the drug development capabilities and the really deep level of resource and expertise that Pfizer can bring to that. And I think I would also add the speed with which we can. We're not usually known, the companies aren't thought of as lightning speed, but we can move programs along quite quickly. And it's really, complementary biology and drug discovery that get performed at Pfizer. And we're really looking to translate that. And again, it's always focused on breakthrough medicines for patients. This is a little roadmap of what our CTI look like. Over 100 projects have been started, and it describes how many are active. And one question, academics always ask us is, can we publish the work that we're doing with you? And certainly there's a strong record from CTI, but I can also say within the rare disease research unit and across the research units, publishing is a key part. And we appreciate that that's, central to any academic's work. And then lastly, we have, a half a dozen programs that are in clinical development that came through this. So really a successful program, and we're excited about this. So I'm going to pause there and take a breath because I've seemingly been just talking to myself in my computer in my room. But we want to spend a couple of minutes just describing the current call for proposals. If that's good with everyone, if no one stops me, I'll keep going. So this describes the current call that we have. And again, we're seeking proposals to advance therapeutics in PKD, in Alport, in FSGS, and IgAN. We have, we're always very strategically focused in how we work. And the key thing about this, again, and I keep stressing that it's novel target concepts and therapeutic strategies. And to slow renal decline. That's really our goal. And we can share this with you. There's a website after this that describes some of the details around this, but we are modality agnostic with the exception of cell therapies. With the success of our COVID vaccine and the lipid nanoparticles delivery method, we're very focused on expanding that capability. And I think that has clear applications for delivery to specifically to the kidney. And I think the last thing that I can say about this is just the sort of operational aspects of it. We're opening the call at the end of next month, and it's going to run for a few weeks. The submission process, we try to make it not an onerous process, just a 2 to 3 page description of the work at a non-confidential level. But Steve and I have our contact information up there because we'd really prefer to have the conversation first and understand what the work is about at a non-confidential level, and if it's a fit for CTI, or maybe it's a fit for one of these many other collaborative processes that we have at Pfizer to work with people. So I'm going to pause there. Steve, anything to add, or Jenna, to the call for proposals?
I think that's pretty clear. We have a template, actually, right, Jenna? Can you hear me?
Yes, we can hear you, Steve.
Okay. Yeah, there's a template that we can provide as well for that 2 to 3 pager where it lays out the rationale and the plan in a non-confidential manner. And then it's always great to connect beforehand, like Aileen said.
Okay, very good. So I am going to stop sharing my video as Kristen asked, and then I'm going to advance the slide. And Steve, you're just going to cue me when you want me to advance the slide.
Okay, thank you. So thanks, Aileen. That was a really great overview of all the different ways to partner with this greater organization that I'm in, where CTI is one part of that. And I also want to thank Josh and Kristen and NephCure for our partnership along the way, as it's been interesting to see the evolution of our collaboration as the story I'm going to talk about as we've moved into the clinic, and hoping to advance and enable novel therapies, getting to rare disease, protein spilling disease patients overall, and we're a small part of that. So it's been a very exciting and really great partnership from my point of view. So you can hit the next slide. So I want to talk about this Robo2 program to provide an example, not just of what we're considering a success story, but also the nature of what was part of their proposal, and then how we work together, and now how we're in the clinic. So in terms of an example from the starting point to where we are now, to provide a little context, if anybody's thinking about a hypothesis or a proposal that's percolating in their lab or in various efforts and other collaborations, et cetera. So we started this program in 2012. I'm based in Cambridge, Massachusetts, at the Pfizer site there, with the Boston Medical Center and Boston University, with a PI named Wenning Liu, who's been an absolute pleasure to work with in this collaborative manner, as well as David Salant, who was the nephrology chair at the time, and many of you know from working on membranous nephropathy and PLA2R. And they came to us with this really unique hypothesis around this SLIT-ROBO pathway, and had some data underlying that with some unique patient genetics from a single patient, animal model data kind of backing up what they believed from that genetics, as well as some work in the cellular and molecular mechanism of how this could be beneficial to podocytes, inhibiting this pathway. And key was the pathway is targetable from a drug perspective. It could be targeted with a biologic in a few different manners, actually, either targeting the receptor or targeting the ligand, FLT2 or Robo2. So we started in 2012, and then we moved into first-in-human clinical studies with the biologic in 2017. And now we're currently enrolling phase 2 study in FSGS, and we have some patients, some data that was presented recently at ASN, and the trial is continuing. So what was unique about the proposal, and I've tried to represent that in the bottom, and it pasted a little funny, I'm sorry about that, where they proposed this pathway where it looks like loss or inhibition of this pathway was beneficial to podocyte structure and function, which is always nice from a therapeutic strategy where we're very good at inhibiting pathways. So we could inhibit this pathway and improve podocyte function and structure, etc. The next slide. So this has resulted in Robo2 FC, which is what we call it, as well as you can see the drug name, the drug number above from Pfizer. And as I said, we're directly targeting the filtration barrier through the podocyte, where loss of function or inhibition of this pathway is renoprotective, we believe reduces proteinuria. And this was started, and I'm going to describe a single observation with a patient who had a translocation disrupting this pathway. And then, as I mentioned, we can target this with the biologic. There were some publications prior to collaborating with Pfizer, as well as we've co-published along the way and presented at various meetings, and I'll highlight that. But the take home is we've generated a molecule which we call a ligand trap, or a decoy receptor, where we have the extracellular domain, a portion of that of Robo2, fused to an IgG1 FC to extend the half-life. So it's almost like an antibody, but a little different, and will titrate and bind the ligands for this receptor, one of which is SLT2. And then we believe this will improve podocyte actin polymerization and adhesion, which then reduces proteinuria in patients. So I want to highlight next slide. And this takes a little bit of time, but this is what Dr. Liu came to us with, where he started with this patient, and this is how he made the leap to moving from kind of a developmental biology story to a therapeutic in adult patients story, where this patient, it's okay, I think I'll start on the left side. It's okay, Aileen. Was mentally disabled actually, and had a chromosome smear in the Developmental Genome Anatomy Project called DGAP. The patient is called DGAP 107. And it was known this patient had a complete translocation between chromosome 3 and chromosome Y. But as a child, went to, had a urinary tract infection, had a scan, and was discovered to have vesical ureteral reflux. So complete backup through the ureters into the kidney on both sides. Why Dr. Liu was initially interested was he wanted to understand what was the gene at this translocation that could have been driving this disorder. And it turns out it's ROBO2 directly at that break point where chromosome 3 moves into chromosome Y right through 5' 3' of ROBO and actually translocates right where ROBO2 binds its ligand SLT2, which is that first IG domain. So he published that work of the importance of the SLT-ROBO pathway in proper patterning and development of the kidney and urinary tract. But they also noticed that while this patient did have some renal decline, he had no proteinuria, despite what could potentially cause a pretty significant secondary FSGS-like condition. And he thought that maybe there's a difference of function of this pathway in development versus the adults, which is quite common across many pathways, where the translocation drove his VUR, but then there's a function that's lost in the adult that's actually protecting him. So it was an amazing flip from Wenning to make that leap with Wenning and Dr. Slant. So next slide. So he then went on to show in the adult, ROBO2 is specifically expressed on the podocyte, on the basal side of the podocyte, and that the ligand SLT2 actually associates with the basement membrane, as well as can be throughout the renal tubules as well, but it's more highly expressed in the glom. And that in that position, they're actually, in that localization, they are accessible to biotherapeutics. So you could target either the receptor or the ligand SLT2. So if you hit the next slide. So he proposed this. And then he also knocked out ROBO. And we've done some of this work in collaboration since the collaboration started, but he had a number of models where he knocked out ROBO2 or either did a podocyte-specific knockout of ROBO2 and showed things like restoration of the structure of the glomerulus, as you can see in the left, in the context of the nephrin knockout, as well as the podocyte process interdigitation, as well as protection of structure and function in various models where you provide stresses to the glom, such as the nephrotoxic serum model, which you see below, where knockout of ROBO2 specifically in the podocyte reduced proteinuria by about 10-fold, as well as and we've built on this together, some mechanistic data where how this could be occurring. So SLT binds ROBO2, and there's actually a lack of a catalytic activity, but these adapter molecules bind to the coil-coil domains within the intracellular domain of ROBO2, and that affects adjacent pathways, such as the nephrin pathway, as well as targeting directly to the actin myosin machinery to affect actin polymerization and adhesion of podocytes to the basement membrane. So some of this we built together as we went, but some of this was part of the initial proposal, kind of validating his thought from that initial patient to a podocyte-driven target, and that loss of this target looks like it could be beneficial. So next slide. So then we did a number of studies. You can just hit through one more. Thank you, Eileen. Oh, back one. We run a few different models, and I just wanted to highlight we generated therapeutics, and I also want to highlight, I forgot one thing I always say, when you're working together, and I think first off, we would have never known about this target without CTI. So we advanced the science much more quickly in the translation to the clinic without this partnership. If we were still just working in a silo, this would have never occurred. And second, you learn along the way from your partner what you've tried and what doesn't work. What technologies, what cell systems, what have you done already that maybe we would have repeated and been spinning our wheels for a long time. And you're working with the other, your partner who really knows, has a little more expertise. You build along the way and you say, look, that's never going to work. And here's why. You just saved us months and months and months, possibly. There's always, then there's the opportunity cost of that, whereas we can work on the things that we think will be productive. So that was really key to then developing different molecules that we knew inhibited this pathway. And the fusion protein Robo2FC, we show here in the passive payment nephritis model, where dosing either early or later on in the model reduces proteinuria and improves podocyte foot process width, as well as the diaphragm density. So we reduced proteinuria, we improved podocyte structure, I guess is ultrastructure, et cetera, and use this type of data to validate that maybe we should see how this does in proteinuric patients. So that's when we started working with NephCure, actually, to help enable our study, which we'll show on the next slide. There's a lot of work in between that slide and here. We had an initial study in healthy volunteers to understand the PK, safety, immunogenicity, et cetera, of Robo2FC. And then we moved into a trial in FSGS patients, which is described here, looking at an open label study where we're dosing 2 different doses of Robo2FC at higher and lower. We're starting with the highest dose to see if we give the molecule the best chance to show an effect, and then actually move to a lower dose to see if that's continued. If we continue with lower doses, it's always great to start the next study at as low as dose as possible to get your desired pharmacological effect. So each patient actually serves as their own control. So it's open label. There's a lead-in period where we follow their proteinuria, et cetera, and then dose Robo2FC for 12 weeks. And then the endpoint is week 13. And then we have a follow-up where we, for safety as well as understanding the PK of the drug. And you can see the inclusion criteria below. So we have significant proteinuria, as well as we're allowing treatment or previous treatment or current treatment with what you would consider standard of care. And I want to highlight up in the right-hand corner as well a biopsy sub-study that we've initiated, and I'm going to show that data. And I want to get through relatively quickly to make sure we have time for questions. But I just wanted to highlight that this initial proposal led to this effort, which I think is quite meaningful. So as I mentioned in the middle, we have interim analyses. So I'm going to share some data where we're looking at either if we're, are we futile, or what kind of an effect are we seeing? So next slide, Aileen. So we did an interim analysis halfway through. We presented this data at at ASN Kidney Week last November, where we looked at our first 9 patients that enrolled, 8 completed. 1 had to discontinue due to non-drug related reasons. And 1 patient actually missed a dose due to COVID travel restrictions. And you can see on the right, I don't want to run through everything. They paid the mean. I'll have the median on the next slide. UPCR of 5.9, which is pretty significant. GFR of 50, and most patients were also on ACEs and ARBs. So next slide. And what we showed was this is a first-in-class, that's what we call it. Aileen made mention. So this is the first type of molecule targeting this pathway. So we call it a first-in-class molecule. And we saw a significant reduction in proteinuria in these patients at the week 13, to the point where it was statistically significant with a much smaller number of patients than we predicted. So we had a 35% reduction at 13 weeks. You can see the mean baseline UPCR was 5.4 grams. The safety profile was very good. No severe or serious adverse events or deaths, etc. And with this work, we continued the study. We're currently enrolling. We've fully enrolled the second half of the cohort, and we're moving into the lower dose cohort where we have some patients who are currently dosed. And we've also included to expand the dosing beyond the 12 weeks, because with this type of pathway, we don't know if we've maximized the effect at 12 weeks. Could the podocytes continue to improve and that number continue to decline? So patients actually have the ability to dose out to 24 weeks. So next slide. So it's exciting. I'll just highlight quickly, we also included the collaboration built within Boston University. We're working with their pathology department. A PI named Joel Henderson is key to our team. Many of you probably know him as well as Wenning and David, where we have a pre and post biopsy. A single patient opted to do this, a major effort, as you can imagine, where we measured podocyte foot process width by quantitative imaging, and we saw a significant improvement in the structure of the podocytes in this single patient. It is an N of 1, but it does, and with all the caveats there, but it gives us reason to believe in our mechanism here. So next slide. And I wanted to highlight, we do publish along the way. We have more papers coming along, either on the mechanisms or on the clinical trial or on new novel biology of the pathway. So as we work together, we generate reagents and technologies, either between Pfizer or the partner, that we then exchange and can build the science together and publish as we go along the way, and publish on the molecules, et cetera, once we're nearing the clinic. So I just wanted to highlight that along the way. We've been very focused on being very science driven. The next slide. That's all I had. I hope that provides, it's also, it's a success story from our point of view, but also the nature of the collaboration, where there's a hypothesis that we think is novel with some reasons to believe that can vary across whatever hypothesis that is. In this case, it was some unique genetics with some animal models. The reason to believe can emerge from multiple types of data. But then there was a vision where we could see a drug discovery program where we build the science along the way and then converge on the clinic. And we've been working together 10 years. So, and it's still continuing, and we're going to be working with Joel on this because we think the work with the biopsies, et cetera, is really important for the field. And I'll stop there. I know we only have 10 minutes, so I'd love to take questions. I hope it's not too much. I hope we didn't confuse with all the different parts of Pfizer, but we have the call for proposal beginning in April, but we'll get that to, we're going to distribute that to various tech transfer offices or directly to departments, et cetera. And then you can reach out to Aileen, myself, and then we'll work with the rest of Pfizer and your side to see if there's a partnership to be had.
Great, thank you. So at this time, if you have questions or comments, please raise your hand. In your Zoom toolbar, there's a function where you can raise your hand. We do have someone. I'm going to Chu Long Huang. If you would like to talk, I've unmuted you. You can unmute and ask your question.
All right. Well, thank you, Stephen, for a wonderful presentation. And I do see the science of a robot too, and what you discussed. I see that project could be very competitive for NIH funding too. So you say the Pfizer is a collaboration, is not a grant. So how does that work? Can someone have a similar project and for NIH funding, and at the same time with collaboration? And how does intellectual property work?
I'll handle that. So you guys hear me okay? So yeah, so most of our labs, all collaborators, obviously have grants, right? So they apply for grants on whatever science they're doing and whatever funding source they'd like. But we add on to that with funding the specific needs for the drug discovery program. And then sometimes we co-publish together with different data from different sources. But no, we don't prohibit anybody from trying to go for grants on the science. And then we co-develop the molecules together, and the IP around the molecules is actually shared with the partner in many cases. It can be. So depending on IP rules. So we don't prohibit at all.
So has not been an issue in terms of institution and NIH and Pfizer. So has worked out very well.
Yeah. Exactly.
Thank you.
Yeah, you're welcome. Great question.
Great question. Anyone else? I have put, you can also enter in any question within the Q&A or the chat function. So feel free to raise your hand. I have a question. Is there opportunity for within CTI? Are you targeting adult type of research-based targets, or are you open to also pediatric?
Either. They
Great.
depend on the disease. For example, Aileen's here, in rare disease. I mean, you can see the focus here, the disease areas, but many of the indications in rare disease affect children.
Yeah, I think it's more than half. So we have a strong focus there.
Great. We do have a question in the Q&A. Lisa says Robo2 is very hopeful for rare renal patients. Did the FSGS patients in the study receive genetic testing to rule out underlying genetic causes of disease? She asked because she's part of Alport Syndrome Foundation and is aware of many Alport patients who are initially misdiagnosed with FSGS.
That's also a great question. So for this strategy, where we thought we had a mechanism that's kind of making your podocytes more resistant to stress, essentially. We didn't require genetic testing to enter, but we can, once the cohorts are enrolled, and try to understand responders and non-responders, et cetera. But for different programs, I mean, you can envision for Alports, we might do the opposite. We would start with genetic testing first. It all depends on the strategy of the disease versus the mechanism that you're testing, as well as trying to recruit in a way that you can generate some data in a relatively reasonable amount of time. Yeah, if that makes sense.
That does.
Thank you.
Nihara asked a question. Are you looking to have a novel small molecule for a validated target for a therapy?
So yeah, so we can partner at any stage of development. Now, if the molecule is fully optimized and ready to go, it might not fall to CTI, but you could partner with Greater Pfizer for sure, with the research units. Or, for example, sometimes an institution could have an initial antibody or an initial lead. It's a little different maybe with small molecules, but then we work together to optimize that, to make it into the best possible therapeutic from a pharmaceutical science perspective, right? To get the molecule with the best drug properties. Sometimes that's where partnerships begin, if that makes sense.
Yeah, thanks, he says. Astrid Wines has a question. Astrid, I'm going to allow you to talk if you don't mind. You can ask your question. You can unmute.
Hey, my question is, most of the research that you've been talked about, and also, the statement of FSGS kind of says to me that your research is mostly targeting disease progression, proteinuria, slowing, something like that. But are you also interested in studying and targeting initial acute podocyte injury? So the initial mechanisms of podocyte injury that will then in some patients and continue to develop FSGS, but not in everybody. So I think what we need to talk about is starting at the beginning of the disease process. So I'm wondering if you're also interested in that, because you're not necessarily stating that in your brochure.
Great question. Yes. So when we're making the brochure, we're just making those bullets as examples, right? In your case, you could envision stratifying patients and really having a target. If you think it's initial cause and then blocking that would obviously, we want these to lead to therapies that would, improve the patient's lives and ameliorate disease. But if that, we could then have a precision medicine approach. It's all going to depend on the hypothesis, right? If there's specific patients who have a specific type of injury driving the podocyte dysfunction, we could target that and specifically target to the right place. Yeah.
Yeah. Okay. And another question in that context, are you potentially equipped to look at RNA vaccine-based therapeutics for, specifically targeting autoimmune diseases? Would that be something you'd be interested in looking at?
Definitely discussing. Yes, I would, Aileen, I would jump in and say, yeah.
Yeah, and I think, and that was what I was the message I was trying to get across on that one slide I showed, where, we're currently focused now in expanding it first to rare and then oncology, but then we think after that, it's obvious that the next path would be our I&I, as we call it, inflammation and immunology department and internal medicine.
Mm-hmm.
And then we also, I'll just highlight, we included, if people have innovative research in enabling, RNA delivery to the kidney, et cetera, would be really, maybe not even having a target or an RNA of choice that they're thinking of expressing, but just enabling that possibility. Because you could imagine that would bring on an entire new spectrum of possible therapeutic possibilities. If you could express your RNA of choice in your cells of choice in the kidney.
Yeah, targeted therapies is a high priority.
It would be tremendous.
Okay.
I hope that answered. Thank you. Thank you.
Anyone else? I don't see hands raised right now, but you are more than welcome. Any comments or questions are welcome. Dr. Cascal says, thank you. Very exciting.
Thank you.
No, thank you. We tried not to do too many acronyms. We're professional acronym players. Yeah, thank you.
Well, this has been terrific. Thank you so much. We really appreciate the opportunity.
And
do you want to, while we're closing up here, do you want to put your emails in the chat? So before we close out, your emails can be readily accessible for any further questions. I know sometimes questions come later when you're pondering all of the information. That's great. Thank you. Wonderful. Well, thank you everyone for being here. We appreciate you. You can definitely reach out to us here at nephcure.org. Josh Tarnoff or myself, we can definitely help disseminate any questions to the Pfizer team. But you can see Miss George Berg, Dr. Berg, did you have any questions? I can allow you to speak if you do. He's put his yeah.
Can I add one thing? We're willing, Aileen and I could speak to your departments, et cetera, if anybody has interest, maybe not just for yourself, but somebody, a colleague, et cetera. We can meet when you're available.
Hi, this is George. I just want to thank you very much for this has been a very interesting hour. We are interested very much in approaches to FSGS. We've actually worked with Astrid Wiens on a common theme before. We're also interested in other types of autoimmunity beyond the kidney. So this has been very helpful and very interesting, and it may very well, I hope, inspire some work in the future together.
Thank you.
It's great. Thank you. I made note of your email. Maybe I can reach out with the call. And we have 2 other calls too that go across therapeutic areas in addition to an RNA call. So maybe there's something there.
That's
All right,
great.
great.
Thank you.
That's great. So thank you everyone for being here. And I'll make sure we capture these emails for you, Aileen and Steven, so you can reach
Great.
back out.
Thank you. Appreciate it.
And I hope everyone has a wonderful day. Thanks again for being here.
Thank you so much, Kristen, Josh. Appreciate it.
Yeah,
We
really great.
Bye-bye.
Bye now.